A-Level Biology 9700, Paper 1, Variant 1, October, November 2025, question number 1 to question number 13. So question number 1 is, in a photomicrograph of magnification 5000, a chloroplast measures 25 millimeter in diameter. What is the actual diameter of the chloroplast? So for this purpose, first of all, we will go with the formula of magnification, which is equal to diameter of photomicrograph divided by actual diameter. And actual diameter will be equal to diameter of photomicrograph divided by magnification. So they have given us magnification and diameter of the chloroplast, which is equal to 25 millimeter. So, we have to change this millimeter or this value into micrometer. For this purpose, 25 will be multiplied with 1000 to get the micrometer. So it will be equal to 25,000 micrometer. Now we will put this value into formula. Actual diameter will be equal to diameter of that photomicrograph, which will be 25,000, and divided by magnification, which is 5,000. So if it will be equal to 5 micrometer, you can calculate it. So D is the correct option. A, B, and C are not the correct options. Question number 2. What is a function of the Golgi bodies? The Golgi bodies or Golgi apparatus act like the post office of the cell. If so, it receives proteins from the rough endoplasmic reticulum and then they modify them. How can they modify? They add sugar chains and making glycoproteins and then packages them into vesicles for secretion. So according to this, the formation of vesicles for endocytosis. So endocytosis is taking things into the cell. Their function is not to taking things into the cell. So this is not the correct option. So B is modification of proteins for secretion. Yes, this is the correct option. Third is synthesis of ATP. This is the function of mitochondria to synthesize the protein. Again, this is not the function of Golgi. Synthesis of polypeptides. Polypeptides are formed by the ribosomes, not by the Golgi body. So B is the correct answer. Now question number 3 is here. And question number 3 is, a cell in the human body is specialized for synthesis and secretion of lipids. Which row shows the organelles required by the cell for synthesis and secretion of lipids? So here you can see which row shows the organelles required by the cell or synthesis and secretion of lipids. So here you can see, first of all, they have mitochondria. The mitochondria is required by the body, which synthesize and transport are active processes that need energy. So mitochondria option will be is correct. Then smooth endoplasmic reticulum. The smooth endoplasmic reticulums are also required because the smooth endoplasmic reticulum is the primary site for lipid synthesis. And then the vesicles are also required because lipids must be packed into the vesicles to be transported to the cell membrane for secretion. So vesicles are also required. So it means all these 3 organelles are required for synthesis and secretion of lipids. So A is the correct option. B, C, and D are not the correct options. Now, question number 4. And question number 4 is the photomicrograph shows part of the cell. Which row is correct? This cell will contain 70S ribosomes. First of all, the first is, so actually, first of all, we will observe this photomicrograph. If this photomicrograph shows the two key eukaryotic organelles, so one is mitochondria and the other is rough endoplasmic reticulum. This is rough endoplasmic reticulum and this is the mitochondria. So now we will go with the options. First of all, the first option is which row is this cell wall will contain 70 S ribosomes. So eukaryotic cells have 80 S ribosomes in the cytoplasm. Mitochondria contain their own 70 S ribosomes. Therefore, this cell will contain 70 S ribosomes. Yes. Then this could be part of the plant cell. So plant cells contain both mitochondria and rough endoplasmic reticulum. So this is also possible. Now the third, the organelles should shown could be used to make antibodies. Antibodies, they are proteins. So the rough endoplasmic reticulum is responsible for synthesizing proteins for secretion, and mitochondria provide the ATP required for this process. So this statement can also be the correct. According to these three statements, the answer is A, because all these three are required or correct. So A is the correct answer. Now, question number 5 is there. And question number 5 is, which statement about viruses is correct? They are cells with their capsid made of phospholipids. Phospholipids, some viruses have a lipid envelope stolen from a host cell, but the capsid itself is not phospholipid or this protein based. So they are not this is not the correct option. They are cells with capsid made of protein. And they are cells with capsid. They are not cells. Viruses are not cells. So they are non-cellular. They are cells with capsid made of protein. So, the capsid is a protective outer shell of the virus and it is made up of protein subunits called capsomers. Again, they are not cells. So again, this is not the correct option. They are particles with a capsid made of phospholipids. Again, this is not good because they are not the particles. They are particles with a capsid made up of protein. Yes, they are particles with capsid made of protein is the correct one because capsid is not made up of phospholipids. Because they are non-cellular, So they are the particles with capsid made of protein is the correct option. Question number 6. Solution X was tested for the presence of non-reducing sugars. It did not contain reducing sugar. Some steps that can be used to test for the presence of biological molecules are listed. So they have given 4 options. So first of all, for non-reducing sugars, we have to add dilute hydrochloric solution to the test tube. In the test tube, which has sample solution. So we will add some dilute hydrochloric acid to the test tube. So it will be the first. Then heat the test tube in a water bath. We will heat the test tube to activate the dilute hydrochloric acid, which will break down the non-reducing sugars into reducing sugar. So after that, we will add some hydrogen carbonate solution to the test tube because it has become acidic by adding dilute hydrochloric acid, which will break down the non-reducing sugar into reducing sugar, but the solution will become acidic. So to neutralize the solution, we will add hydrogen carbonate, which is an alkali. So it will be the third, and then we will add Benedict solution to the test tube. So it will be the fourth. Now after that, again we have to heat to for Benedict test whether now the non-reducing sugar which has been changed into the reducing sugar. So we will heat it again. So again it will be the at 5. So 2, 4, 3, 1, and so C is the correct option. Now question number 7 is the diagram shows the structure of part of biological molecule. So here you can see a biological structure of part of biological molecule. So we will go towards the options. Which statement about these biological molecules are correct? You have seen that it has monomers joined by ester bonds. These are not monomers. You can see. So it is a polymer made of multiple sugar rings which are linked together. So statement 1 is not correct. Then in monomers or sugar molecules when are joined together, they are not with the ester bonds. There are glycosidic bonds. So this is not correct option. Second, it is a macromolecule. Yes, this is correct. Very large molecules such as polymer, since this shows a long chain of repeating units, fits. So this is correct. It is a polysaccharide. Yes, the individual units are monosaccharides. When many of these are linked together, they form a polysaccharide. They are with the help of glycosidic bonds. So 2 and 3 statements are correct, and It means D is the correct option. Now question number 8 is there. And question number 8 is, which diagram has a covalent bond that joins monomers together to form a biological polymer in eukaryotes. So here you can see the A shows an ionic interaction. You can see at plus and minus. So these plus and minus show ionic bond formation is there. And in HO, this is hydrogen bond. And at C, you can see an ester bond. And at D, you can see a phosphodiester bond, which is the backbone of the nucleic acid formed between nucleotides. So here you can say phosphodiester bond. So D is the correct option. So covalent bond is there in D only. C, B, and A are not the correct options. Now, question number 9. Which feature of storage polysaccharide means they do not change the water potential of cells? So, here the question 9 is, they are easily hydrolyzed. They are easily hydrolyzed. Hydrolyzed means they are broken down. When they will break down, when they will hydrolyze, they will change the water potential. So A is not the correct option. Now B is, they are compact. They are compact. Again, they are not, you can say, this is not the correct option again. So they will not affect the water potential. They are insoluble. This is the correct option because they will not dissolve in the cytoplasm. So they will not change the water potential of the cells. So C is the correct option. They are branched molecules. Again, this is not the correct option. C is the correct option. When they are branched, you can see they will easily break down. Their bonds will be broken down and then they will change the water potential of the cells. Only C, when they are insoluble, they will not change because they are not dissolved, they have no osmotic effect and they do not pull water into the cells, so which prevents the cell from swelling or bursting. So C is the correct option. Now question number 10 is there. And question number 10 is, which molecule contain at least 4 double bonds? So here, first of all, hemoglobin and collagen. If we observe their structure, then we can find out this carbon and oxygen, they want carbonyl or other double bonds in these molecules. So a saturated triglyceride has three ester bonds, meaning it has exactly three carbon-oxygen double bonds. Since it is saturated, there are no carbon-to-carbon double bonds in the fatty acid tails. Therefore, it has fewer than four. So triglyceride, no, only collagen and hemoglobin will have. So collagen are large proteins. Proteins are made up of long chains, amino acids joined by peptide bonds. Every single peptide bond contains a carbon-oxygen double bond. Since these proteins consist of hundreds of amino acids, they have they have hundreds of double bonds. So we need the area that include collagen and hemoglobin that excludes the saturated triglycerides. So on the Venn diagram, that section is B. So first, this hemoglobin and collagen. So according to this, B is the correct option. Now question number 11 is, in some people, a mutation can affect the beta chains in their hemoglobin molecules. As a result, polar amino acids on the outer surfaces of the chains are replaced by the non-polar amino acids. What will be the effect on hemoglobin? Hemoglobin will become, first of all, the beta chains in their hemoglobin, polar amino acids on the outer surface of the chain replaced by non-polar amino acids. And here you can see that hemoglobin is a globular protein that stays soluble in the blood because its polar amino acids are on the outside, so interacting with water. So if we replace these with non-polar amino acids, which will make the surface hydrophobic, so these non-polar patches will clump together to avoid water, making the protein less soluble. This is actually the mechanism behind sickle cell anemia. So according to this logic, hemoglobin will be less soluble. It will become more soluble? No. Due to hydrophobic nature, hydrophobic interactions between amino acids will decrease? No, they will increase. They will not decrease, they will increase. Most hydrogen bonds will form in they will not form, they will break down in this way. So it will become less soluble. A is the correct option. Now question number 12 is there. The graph shows the effect of substrate concentration on the enzyme catalyzed reactions with and without a competitive inhibitor. What is the effect of the competitive inhibitor on V maximum and so you can see the graph which dotted lines is with inhibitor and straight line is without inhibitor. So what is the effect of the competitive inhibitor on Vmax and Michael-Menten constant? So if we draw the line, so this is the maximum velocity and this is the half velocity. So the half Vmax here will be the Km. So you can see V maximum decreases and K maximum decreases. If V maximum is decreasing, the K maximum is increasing. So A is not the correct option. V maximum stays the same and Km decreasing. Km is increasing, not decreasing. You can see it is increasing. So V maximum stays the same and K maximum increases. You can see it is without inhibitor and this is with inhibitor. So you can see this is increasing. So C is the V maximum decreases and V maximum will remains the same. So this is not the correct. Only C is the correct option. Now question number 13 is there. First of all, graph shows energy changes in a chemical reaction. Progress of changes, what is the activation energy when an enzyme is added? First of all, you know arrow 1 and 2. This is the arrow 1 and arrow 2. The energy required to reach the transition state with an enzyme, it is, you can see the lower peak. So the third one, the net energy released, the net energy release is 3. And you can see the activation energy is the distance from the starting energy of the reactants to the top of the curve. It is the distance from the starting energy of the reactant. So starting energy of the reactant is this one and reaching at the top to this one. So with an enzyme, this peak is lowered. So this is the lowered peak and this is the without enzyme. So this is with enzyme. It is 2. So 2 is B and A, C and D are not the correct options. So it was all about from question number 1 to 13. Please like, share and subscribe my channel to watch more videos on this paper and on other papers. And thanks for watching.